Although the proofreading system employed by DNA polymerase is impressive, the integrity and fidelity of DNA is still compromised by other factors. This necessitates multiple repair systems to correct incorporation errors or DNA damage that occurs after the replication process is complete. Mutagens are agents that cause mutations to DNA. One very common mutagen is ultraviolet light, which creates what are known as pyrimidine dimers, illustrated below. The light induces the formation of a cyclobutyl ring between the pi electrons from two carbons on adjacent pyrimidine residues. This structure distorts the DNA double helix. Ionizing radiation, chemical agents, or free radicals can lead to a break in the DNA backbone through destruction of the sugar ring, as shown below. As there are different ways in which the DNA might be damaged or errors introduced, so there are different repair mechanisms that function to reverse or correct the mutation. The first we will consider is mismatch repair. This occurs when the wrong nucleotide has been incorporated into the DNA chain, resulting in two incorrectly paired bases. Enzymes must recognize the distortion caused by the mismatch, remove the wrong nucleotide, and replace it with the correct one. However, since there are two strands, the challenge is to determine in which of these strands the error has occurred. The key to this mechanism is the fact that DNA contains specific sites in which one of the bases is methylated after replication has occurred. Shortly after replication, though, only the original parent strand is methylated, as illustrated below by the blue strand. The new daughter strand, the red strand, has not yet been methylated, and this is the point of distinction between them. Mute proteins recognize the mismatched pair and form a loop with the methylation site. The unmethylated daughter strand is recognized as containing the incorrect nucleotide. Now an exonuclease removes a segment of the DNA containing the mismatched base, as shown below, and DNA polymerase uses the original parent strand as the template in order to fill in the missing nucleotides with the correct sequence. Note, however, that this mechanism only works if the new strand of DNA has not yet been methylated, shortly after replication has been completed.